name is Nathan. I'm a sixth-year PhD student in Dr. Paulson's lab. And what I study is basically the intersection between structural biology and systems biology. So just as an introduction, this is kind of going to be a sidestep from what you guys have been learning this quarter. We kind of consider structural biology as, we think of it as an orthogonal data set to everything that you guys have been learning about omics and putting networks together and simulating with them. These tell you more about the actual nodes in your network. So these are the machines that carry out the metabolic reactions in your cell. And it's important to understand what they look like and how they function. And so structure can answer that question. And so what the topics will be split up into 2 lectures. So today I'll be giving you guys some real basic biochemistry 101. So the basics of protein structures and how you can measure experimentally properties of them and compute with them in silico. And then on Tuesday, what we'll be talking about is more of the work that I've been doing and some other people in the lab, where we've constructed and reconstructed all the structures that are necessary to simulate with in a metabolic network. And we'll dive into all the applications that have been done in the last, I think, 10 or so years from our lab. All right, so just to ease you into this topic, here's a network. Here's a network of something. And in this case, it's the network of interactions between residues in a protein structure. And so it's really a simple concept, I think, to understand. Protein structures are simply systems at a really small level. It's at the atomic level. And the scale we're talking about is on the scale of angstroms. So that's 10 to the negative 10 meters, a little smaller than a nanometer. And the length of and just to get you situated, the length of an E. coli cell is about 20,000 angstroms, and the average diameter of a protein is 50 angstroms. So you can imagine that there's going to be a lot of proteins in a cell. And comparatively, the study of protein structures is a much older field of study compared to these omics technologies, which are really recent. You know, these are like in the last 20 years have developed, but protein structures have been studied since the 50s, and the techniques to understand their structure have been developed since like the early 1900s. So these kinds of techniques have been used and have been developed and improved upon for a long time now. And so that brings us to the question, you know, why should we study structures? Why should we understand what they look like? And an analogy here is kind of like if you're thinking of a system as a car and you're trying to reconstruct your car and model it in silico, it would be important to understand the components of that car. Say, why is this engine in this Honda Civic so different from this engine in this truck? And understanding the structure, how it's built, the components inside of it, that gives you insight into how it will treat the entire system itself. So understanding important components of the cell in detail can help you reconstruct networks. It can help you understand interactions such as like pharmacology interactions, like when you take a drug, they interact with your proteins and they do something to you to cause an effect. There's also a whole field of enzyme kinetics, so understanding the rates of reactions that are happening. Structure can help you understand what reaction is actually happening and how fast it occurs. And there's another whole topic of regulation. So proteins are involved in major regulatory networks within your cell, and they, of course, carry out all the reactions. So having a certain amount of protein is important to actually carry out everything that's happening in your cell. So here's just a figure showing where proteins fit in within this paradigm of genes to whole bodies, and that's where they are. So they're what's transcribed by your genes. So here's just a really brief history, as brief as I can make it for you guys. And so, as I mentioned, in the early 1900s, the techniques to determine structure were starting to be studied, and these techniques were mainly X-ray crystallography and NMR. And in the 50s, a bunch of stuff happened. So, as you all probably know, the X-ray structure of DNA was solved in 1953, and a couple years after that, the X-ray structure of myoglobin was solved. So myoglobin is an oxygen-carrying protein in your body. And a couple years after that, hemoglobin was solved. So these were huge discoveries because these were really the first macromolecules that people could see in detail and start to understand, how they work because of what they could see. So in the 70s, people were starting to crystallize structures and determine structures even more. So the protein data bank was established, and this has become the main repository for structural information today. And also in the 70s, people were able to finally visualize these things that they were solving on their computer. So computer graphics became possible instead of having to build ball and stick models of DNA or protein in your office or something. And what that allowed us to do was actually simulate with these proteins. So they began to do research in how to place the protein in your computer, put it in a box of simulated water, and visualize how it moved. And so that has been developed until today as well. And some recent developments in structural determination have been in determining much larger structures. So here's like a virus capsid as compared to these tiny proteins here. And you can see all of it at once now, instead of just determining pieces of it. And another thing that's been happening recently is advances in membrane protein determination. And so membrane proteins, of course, are very different. They behave very differently than proteins floating around in solution. And you need to have many different techniques to crystallize and determine their structure as well. And also recently there have been these efforts called structural genomics, and structural genomics is really just trying to crystallize and determine all the structures in a whole genome. And this has allowed us to finally use these structures on a genome scale, for example, in our metabolic networks. And so that's the reason I'm here. And that's, there's a lot of things I think that are possible to be studied still. And so and we'll talk about them in these slides. So today there are over 135,000 structures available in the protein data bank. When I first started grad school, I was saying 80,000. So that's how much the number has increased just in the last couple of years. So this stuff may be very basic to you, or it may be new to you depending on who you are, but I'm just gonna go over the very basics of protein structure. So these, we'll be talking about the building blocks of structures, which are amino acids and their biochemical properties, which may influence a whole network. The assembly of these amino acids using peptide bonds into the final structure. The patterns that you see with structures, and then finally we'll look at how you experimentally determine what a structure looks like, or how you predict it computationally, and we'll talk a little bit about the function and the regulations that occur with structures. So it's a little washed out, but I mean, these are just the 20 amino acids, and this should be pretty familiar to most people. Obviously, there are differences in size and shape of amino acids, which can influence what a protein structure looks like in the end. And other main properties that you need to consider are the hydrophobicity. So do I like water or do I not like water? And that will influence your friends, basically. That will influence if you hang out with your friends that also like water inside a protein, or outside a protein, or don't like water inside a protein. And the charge and polarity goes along with that concept, as well as reactivity. So some amino acids don't react with anything at all, and some, like cysteine, or with the active sulfur residue, those react with a lot of different kinds of molecules, and those are important to consider, and they're usually found inside a protein at an active site that reacts with a metabolite or something like that. So these kinds of properties can be considered at an amino acid level, but they can also be considered on a whole protein structure level, and you'll see that that amino acids will group together based on these properties to define the final shape that you see. So that's just something to keep in mind because what we can do is we can calculate these properties proteome-wide, see how they're distributed throughout protein structures and throughout a metabolic network, and we can use that to understand how that may impact reactions that are happening. So this should also be pretty basic, hopefully. So obviously the genome it encodes specifically for amino acids, and what happens is these amino acids will assemble together and they form peptide bonds. So these are covalent, strong bonds that are forming, and so that is, and researchers have developed these terms. So there's primary structure, secondary structure, tertiary structure, and quaternary structure. And these are just terms to help you organize your thoughts, basically. So primary structure is really just a sequence, and you can think of these, you can organize these as in terms of the bonds that are being made. So primary structure really just considers that the peptide bonds, the covalent bonds, are strongly linking your amino acids together. Secondary structure starts to think about the patterns that are being formed that you see in structures all the time. So these are really just a couple, which are the alpha helix and beta sheets. And so that's an alpha helix, that's a beta sheet. And the tertiary structure is what you can consider as the final structure for your protein sequence encoded in a genome. And that is your final folded blob of stuff that that potentially carries a reaction out. Or this may not be an active protein all the time because it may have to assemble or repeat itself multiple times to form a quaternary structure, so a more complex structure, either composed of your protein or multiple proteins coming together all at once. And these are characterized, tertiary structures and quaternary structures are characterized by non-covalent, weaker bonds, and so they're not permanent. They can detach from each other and move around. And so another thing that is commonly studied in structural biology are the patterns that we see. So these are, secondary structures are patterns, but you can see even more patterns. And people throw around words like motifs, domains, folds, and families, superfamilies, I don't know. So it gets a little confusing sometimes, so I try to, the way I understand it is that motifs are really defined as secondary structure motifs. So these are small repeating units of alpha helices and beta sheets that you commonly see. So an example would be a helix turn helix motif, which binds to DNA a lot. And so here's a alpha beta loop. And so the terms get confusing because they use these same terms to define all these patterns. And you may not know if they're talking about a motif or a domain or a family. But as long as you look at the structure, it's easier to understand. Another way to think about it is the length of a pattern. So a motif is usually around 10 to 40 residues. And so next up is a domain. A domain is a bigger thing. And a domain can be characterized by if it's independently stable. So for example, a protein sequence may have two domains. One domain, you can just cut it off of that protein structure and it can roam around by itself. It's independently stable. And what people do is, engineers and even biology itself does, is they rearrange these domains to be used as functional units throughout a metabolic network or through evolution. So that's an interesting concept to think about because that helps you break down the structural space into functional units that you can think about. So these are usually around 50 to 200 residues long. And these are just guidelines. I mean, you can have domains that are very long or very short as well. And so from the top down, they have also, researchers have also tried to classify these patterns into even more nomenclature. And so this nomenclature, you'll see things like classes, folds, and superfamilies. And so a way to think about this is how the kingdom of life is classified. So you have kingdoms, phyla, genuses, species. This is kind of the same thing here with protein structures. And I'm not going to talk too much about this, but it's a whole field of study of structural evolution. And you can learn a lot from this because, as we'll talk about later, structure is more conserved than sequence. And so if you can understand the distribution of domains throughout evolutionary history that can tell you about how evolution has occurred. So that leads us to the structure itself, and these are illustrations from David Goodsell. And so David Goodsell is a biochemist here at the at the Protein Data Bank and at Scripps, and he's a really interesting guy. He draws these things by hand. I don't know if these are drawn by hand, but he draws these whole cell images by hand. So the first title slide that I had on the left there was something. He draws every single protein by hand. And he watercolors them, and he puts them up on the wall, and they look amazing. So that's just it's just a really cool, I think thing. He's a cool eccentric guy, I think. So there's a video on YouTube that describes his process. It's really neat to understand how he does that. So once you have the structure, that allows you can just look at them. You can obviously just looking at them tells you that these proteins should have different functions just based on how they look. And I just want to give a brief introduction to how we actually determine what they look like experimentally. And so the main methods that we use are so I'll talk about the main methods in the next slide. So the usual process is that we need to express our protein in a model organism. And then we need to purify it, so we need to have a pure sample. So you guys can obviously, I think, understand that can be a long process. So that's all done in the wet lab, and you have to have just your protein in a test tube and use that to send it through your structural determination method. And so we'll talk about those, which are, there are three main methods used, which are X-ray crystallography, NMR, and cryo-EM. And so once you do that, it spits out a model of what this protein should look like based on what you see with these experiments. And you can fit your protein sequence of interest into this model, and then that's your structure. And so that's when you would deposit it into the protein data bank. So this whole process, like in the 90s, would have gotten you a Science or Nature paper because it took so much work and you got so much information out of it by doing these experiments. But now you have robots that can do a lot of this, so you can't get a Nature paper anymore, unfortunately, unless you're doing something really important, like it's a viral protein that you found a drug that bound to it or something, and you can explain the mechanism behind it. Yeah. So here's just a quote. A structure gives a complete, unambiguous identification of the compound, connectivity of atoms, inter and intramolecular interactions, stereochemistry, and basically a total molecular characterization. So that's what you get with structures. So here are the methods that I was talking about in terms of the structure determination part. And these three methods are pretty much the main methods used. And X-ray crystallography is by and far the dominant method in the field. And so with these methods, again, you have to have a pure protein sample. And I'm just going to give an analogy for X-ray crystallography because I worked on it as an undergrad. And the way I understood it was you have an analogy here is that when you're looking through a light microscope, light bounces off what you're looking at. It gets sent through a lens and then gets received through your eyes, and you get a magnified image in your brain that processes the light rays that bounce off of it. An analogy here is that we're using X-rays instead of light rays because of the smaller or the longer wavelength. And what that allows you to do is it will bounce off these protein molecules, and instead of going into your eyes and probably making you blind, it goes into a computer that receives these signals. And your computer processes the image for you. And what it spits back out for you are these dots, so you can barely see them here, but they're called diffraction patterns. And what happens next is, instead of your brain processing the image, you have to sit there and model your protein based on these patterns that are spit back at you. And from that, you get an atomic model. So that's the main process that has been used to determine protein structures in the last 100 years or so. And the other methods are NMR and cryo-EM. So cryo-EM is somewhat similar to X-ray crystallography in that you use electron microscopy to get patterns sent back at you. NMR is a totally different concept where you try to you exploit the characteristics of excited atoms, and you send different wavelengths of light to them, and you see how excited they are, and you can measure the distances between these excited atoms and get a model based on that. So I'm not an expert on NMR, but that gives you different information actually, because that can tell you kind of about the movement of a protein. But it also gives you a structure as well. So here's just a comparison of these methods. X-ray crystallography is the main method because it's well developed. You can crystallize almost any size of protein, so small or large, and you can get a very high resolution. And so resolution, I'll explain in the next slide, but you can think of it as a computer resolution. You know, the higher resolution, the better. NMR, you're limited in terms of the size of objects you can study, but you still get pretty high quality information. Cryo-EM is an interesting development in the field, and in the last 10 years or so, it's been said that this is going to become the main method because it's easier than X-ray crystallography. You don't have to have a crystal of your sample, and you're able to just use an electron microscope to see immediately what your protein sample looks like. And so there's a lot of development being done here. This is kind of the next big thing in structural biology. So they're trying to get the resolution higher for these proteins that they send through cryo-EM. Here's just a note on what resolution is. And so as you see, these meshes here represent what you get back from the computer if you do something like X-ray crystallography. And what you need to do then is try to fit your protein residues, your amino acids, into these meshes. So the lower resolution you get is a higher number, actually, because it's measured in angstroms. So it just tells you the smallest length the smallest length that you can see in your model. So a smaller number is better because you can see smaller lengths, so you can see more chemical bonds, basically. And this is important, just like any other kind of biological data set, because you need to understand the quality of information that you're getting back. And just like a genome assembly may be incomplete, your protein structure model may also be incomplete. You may be missing parts of it because your model just wasn't sure what it looked like. You may be unsure if this residue is oriented this way or that way because it's just unclear in these models. And yeah, these are just things to consider when you start using structures on a genome scale. So if you don't have a structure, there are methods to predict what it looks like. And these methods are called homology modeling and ab initio modeling. So homology modeling is an easy concept to understand. You look for a sequence that's similar to what you are wanting to understand. You look for if it has a structure available, and you model it based on that structure. And you can do that for different parts of the protein. Say, for instance, it's split up into domains. You can model those different domains and piece them together using these methods. And the other way is if you don't have a template, you just kind of have to guess it, and you throw it into a computer simulation and you wait for it to fold. And this is an incredibly hard problem to solve, obviously, because there are so many different, you can see in this video that there's so many different ways that it can fold, and you don't know if it's reached like a local energy minimum or if it's like the full energy minimum, but then you also don't know if a structure should be at the full energy minimum or the local energy minimum. So there's a lot of questions that cannot be answered, really. In computational terms, that's a NP hard problem, if anyone is a computer scientist here. And yeah, so that's a whole other area of research, but it's not something that I work on. And so here's just a pie chart from, I think, 2013. So this is a lot better now. This is the distribution of the E. coli proteome solved. This is the solved portion. This is the able to be modeled easily or of medium difficulty. And this is a hard to be modeled portion. So since this was like 5 years ago, this has gotten a lot better. I would say the solved portion has probably gone up to 30 or 40%. And the easy and medium ones have probably increased as well. So what that means for us is that we can start to use structures on a system scale, so we can start to represent the nodes in our metabolic network. Instead of just a node, they can be represented by what they actually look like. So here's just a list of databases that contain a lot of this information that I've just talked about. And so luckily, the Protein Data Bank remains the only source of protein structures, and you can just go there and download that information. There's a lot of protein annotation information on places like UniProt, the catalytic site atlas, which tells you where the catalytic sites are on a protein. And these are all protein family and domain annotation databases. And there's databases of homology models, enzyme kinetics, and of course pathways. And so we've worked with the Protein Data Bank to incorporate a pathway viewer into their protein pages. And so that's just cool because now we can, instead of getting systems biologists to start thinking about using structures, we can get structural biologists to think about how their protein fits in within a bigger metabolic network or something like that. Finally, so once you have your structure, then you can start thinking about how that relates to function. So what do proteins do? They carry out reactions in your network. They transport things. They regulate functions. They act as cell signaling receptors. And they not only have structure, but they provide structure to a cell. And beyond this, you can modify a protein's function in a couple different ways. There's allosteric interactions, where if you, the easiest thing to understand here is if a drug binds to this protein, it will, not at the binding site, but at another site, it may inhibit a molecule being catalyzed at the catalytic site. So that's an allosteric interaction, and it's kind of a regulation on a protein's function. And you can also have post-translational modifications. So these are even more things that you can do to a protein structure to either modify its function or structure, which can inactivate it in different ways and potentially deactivate the rest of a metabolic network, or of a pathway, sorry. So these are things, I think there's a lot of potential to study this kind of stuff at a system scale, and that's not something that I've looked at. Just because there's, we've been talking about all these other things and trying to integrate those kinds of things, and then these are even more things that can happen to a structure to impact it, and it's just, you need to consider everything else before you can get to this. So that, I think there's a lot of things that can be done here in terms of how they regulate a metabolic network. So here are just some, I think, kind of rules of thumb about sequences and structures and their function. So the holy grail would be that if you could predict structure from sequence and then predict function from the structure. But obviously that's not going to be possible for a long time. But there are some rules of thumb that you can use to understand this relationship. And so, as I mentioned before, structure is much more conserved than sequence. And what that means is that you can have as low as 30% sequence identity between a protein in your body, say, compared to a protein in bacteria, and they can still carry out the same function and they can still look the same. But that isn't to say that you can have similar structures that can have very different functions. So that just makes it more complex.